for the introduction, and thank you all for being here. This video shows the entire brain of a fruit fly. It's less than a millimeter in size, but it still contains reconstructions of about 130,000 neurons, as well as many million synapses or connections between these neurons. And these sorts of connectomic measurements are now being increasingly available, and we've already mapped, for example, the entire brains of the worm C. elegans, of the fruit fly Drosophila melanogaster, which you can see here, of the larval zebrafish, and of parts of the mouse brain. For computational neuroscience, these kinds of data are really a dream come true. They provide us with exactly the connectivity matrix of how of which neuron A connects to which neuron B. And on top of this, we also get information of the morphology or of the shape of each of these neurons, such that we can infer where exactly neuron A connects to neuron B. And on top of that, through decades of research, we also have a lot of genetic information. So we know which kinds of proteins or ion channels are expressed in which types of cell, based on the cell type, for example. And that lets us know a little bit about how individual neurons will react to input currents. Today, I'm going to talk about how we can use such measurements, such biophysical constraints, to build neural networks that are a little bit less artificial and that really respect the known empirical facts about what's actually going on in biological brains, for example, in the fruit fly, in larval zebrafish, or in mice. Now, I think it's really tempting to just say, well, why don't we just turn it on, right? We know exactly the connectivity matrix of each of these neurons. We sort of know what they're doing. Why don't we just put this into a simulation and let it run? Well, while we know so many things about brains and about a lot of neural networks, we still don't know many, many other things. For example, in each of these neurons, there would be ion channels that govern how a particular cell reacts to input currents. And while we might know which ion channels exist in what kinds of cell types, we certainly do not know how many ion channels are present in each cell. But that really impacts the response of the neuron. Similarly, we know if neuron A or whether neuron A connects to neuron B, but we only have a very, very rudimentary guess of how strong such connections or synapses really are. And that makes it challenging to build simulations that actually do stuff, that actually match data or that actually perform computations, that actually make a fly walk around or fly around. So how do we fix this? Well, one option that we're going to take in this talk is to assemble all the things about biological brains that we do know into a biophysical simulation, and to then infer the remaining things that we cannot measure with inference, such that the simulation matches activity measurements or performs computational tasks. So you could imagine that, for example, for some neural systems, we actually have calcium recordings that measure the activity in individual cells, and we might want to tune the free parameters such that the simulation matches these data. Or in other cases, we might have a good guess of what the system is trying to do. For example, if it's a visual system, it might aim to classify patterns or to predict visual motion. And we can frame this as a machine learning task and then train the biophysical system or train the parameters of the biophysical system such that the simulation actually performs this computational task. This seems very principled, right? We just assemble everything we know into a simulation and then perform inference for the stuff that we don't know. But again, that's not so easy. And in particular, for biophysical simulations, but for many, many other simulations in the sciences or in engineering, performing inference is hard. And that's because the likelihood, which describes the forward model or the forward simulation, has some issues attached. You can imagine such a big brain simulation. Running this simulation can actually take many seconds, minutes, or even hours to run just once. And basically any kind of inference technique usually requires many repeated evaluations of the likelihood. In addition, a single cell can even already have dozens or hundreds of free parameters. And thereby network simulations can have hundreds, thousands, tens of thousands, or millions of free parameters, which makes it hard to find any kind of parameter set that even roughly matches the data. And thirdly, in many cases, the simulators are stochastic, for example, to model variability in biological systems. And in those cases, it can happen that the likelihood cannot even be evaluated at all, which makes approaches such as maximum likelihood estimation or Bayesian inference challenging. Today, I'll talk about two approaches to overcome these limitations. I'll talk about simulation-based Bayesian inference with neural networks and about differentiable simulation. I'll start with simulation-based Bayesian inference. As the name suggests, these methods aim to perform Bayesian inference, which itself aims to compute the posterior distribution, the probability distribution over parameters denoted by theta, given measurements denoted XO. And with Bayes' rule, we know that the posterior is proportional to the product of likelihood and prior. Now, as I just said, the likelihood can have strings attached, right? It can be very expensive to evaluate in cases where the model just takes a long time to run. And in some cases, it cannot even be evaluated at all. And in others, the gradient might not be available, either because the simulator's been written in a programming language that doesn't provide us with automatic differentiation, or because it has some sorts of discrete states that just set the gradient to 0. Simulation-based inference aims to make Bayesian inference possible for these kinds of models. And one of the popular techniques for this is called neural posterior estimation, which I'm going to describe now. Let's assume you have some kind of a computational model or a simulator, for example, one in neuroscience that models the change in voltage over time. And let's say we also have a kind of measurement of the actual activity of a neuron at the bottom right here. Given this theory or simulator and the measurement, we're aiming to infer two parameters, in this case, sodium and potassium conductance. And with neural posterior estimation, we do this as follows. We first sample parameters from the prior and run the simulator so as to generate a simulated trace. We don't do this just once, but we do it many times, thereby generating a database of parameters and corresponding simulation outputs. And then we train a neural network to invert this mapping. It's a neural network that takes as input the simulation outputs and predicts the parameters that generated it. Crucially, because we do all of this on simulated data, this is now a supervised learning problem, and we've become really, really good at solving supervised learning problems with just standard AI. In order to recover the posterior and not just a point estimate, we're using conditional generative modeling here and thereby predict a full distribution over parameters that match the data. After training, we can then query this trained neural network at any kind of data, and it's thereby going to predict the posterior distribution over parameters. This strategy has a couple of really big advantages, especially for simulation-based models. First off, it fully decouples simulation from training and inference. So if your simulator is, for example, written in C++ or Fortran, no problem. Run it there, load the simulations in Python, and train your inference model there, and then perform inference. In addition, this allows us to perform simulations fully in parallel. Right? There's no sequential steps at all. If you have 10,000 CPU cores, you can parallelize your 10,000 simulations on those CPU cores and just simulate there. Thirdly, this is extremely flexible. It works for any kind of black box simulator for which we can run simulations given parameters. There's no gradients involved, there's no likelihood evaluations involved. All you have to be able to do is to run your simulator. And thirdly, this amortizes inference. Fourth, this amortizes inference, meaning that once this network is trained, we can query it at any kind of data, and it's immediately going to predict the posterior distribution without any further simulations or without retraining. And that's really useful in cases where we want to perform inference in real-time scenarios where data comes in and we immediately want to know the posterior. Over the past years, we've extended neural posterior estimation in many, many ways in order to improve its flexibility, its robustness, its interpretability, its accuracy, and its simulation efficiency. And now just recently, we've written a guide that allows users to navigate this landscape of existing methods and helps them in choosing the method that actually is best fitted to particular problems. Over the past years, we've also developed and maintained the SBI toolbox, which implements many of these methods and makes it easy for domain scientists to just provide data, that is the simulation outputs, and we train the neural network and perform inference by sampling from the generative model. And over the past years, the SBI toolbox has become really popular and has helped people perform inference in models as wide as particle physics, astrophysics, biophysics, neuroscience, economics, biology, and genetics. But I do actually now want to get back to the original motivation, namely to neuroscience. So how can we use this to build biological systems? This is a cell from mouse cortex, a so-called layer 5 pyramidal cell. And we aim to use neural posterior estimation to infer 23 parameters of the cell, for example, the densities of different ion channels, such that the model reproduces features of voltage traces, which are responses of this neuron to different input stimuli. When sampling these parameters at random, the simulation, here one parameter set in each row in black, doesn't match the actual data in orange at all. We then used neural posterior estimation, or rather a slightly modified version that we developed in order to be more robust and simulation efficient, in order to infer the posterior. And when then sampling from those simulating those posterior samples, we found that the simulations closely matched the features of the actual empirical data. Now that's already cool, right? It provides us with simulations that match the data. But we can now also go in and actually analyze parameter space. So here I'm showing 10 parameters out of those 20. And on the diagonals here, you can see the one-dimensional marginal distributions of the posterior. And we can now, for example, study which parameters have to be very tightly tuned. For example, this one has to be really tuned to very low values, whereas other parameters are relatively sloppy and can vary on broad ranges while still reproducing the data. Now, that's already interesting because it sort of tells us how single cells process information. But getting the posterior for single cells can also be really useful for network simulations later on. For example, we could imagine that we assemble single cells into a network, and we can then already initialize the network simulations with the maximum a posteriori estimate for these single cells, such that the network simulation already gets initialized in a region where the network activity where the single cell activity is somewhat healthy. And in addition, a nice property of neural posterior estimation is that it returns a posterior which can also be evaluated at any kind of parameter set. And this we could use, for example, to regularize network simulations such that if we optimize the entire network, the activity doesn't go vastly outside of what we know from these single cell measurements. All of this, however, is still single cell. So in the second part of this talk, I do actually want to move towards network simulations. And this we're going to do with differentiable simulation. Now, in particular, the simulation-based inference approaches that I've just discussed have two really big limitations. The first one is that they struggle to scale to models with many parameters. They routinely work for models with, let's say, 5 to 10 parameters, sometimes dozens of parameters, but rarely with, let's say, 100 parameters. And in addition, they also struggle to scale to very large data sets. But that's kind of exactly what we would want for biophysical models, right? We would want to infer possibly hundreds of thousands of parameters of large neural systems, their synapses, their connection strengths, single cell properties. And we would like to fit these parameters such that the network matches large-scale recordings or performs computational tasks. Now, interestingly, gradient descent is very, very good at exactly those two things. It can scale very well to models with billions of parameters and to gigantic data sets, basically the entire internet. And so what we wanted to do was to assemble these kind of network simulations and then train their parameters with gradient descent. Unfortunately, that wasn't really possible for a very simple practical reason, namely that these kinds of simulators that define these biophysical models were written in C and therefore do not allow automatic differentiation. To overcome this, we've sat down and we've re-implemented these kinds of simulators in JAX. And we call the resulting toolbox JAXLY, which stands for JAX and Hodgkin-Huxley. And for those of you familiar with it, you can think of JAXLY like a clone of the neuron simulator, but fully written in JAX. Thereby, JAXLY allows to do exactly what other simulators for biophysical modeling allow to do, namely to insert ion channels and to model the exact voltage in different neurons at full biophysical detail and at the single branch level. But unlike these simulators, we now get all these beautiful features of JAX. For example, to compute the gradient with respect to single cell properties or to synaptic properties or even to morphological properties of single cells. And secondly, because it's JAX, people have really carefully thought about how we can elegantly parallelize code, for example, with the VMAP. And all of these things now also apply to JAXLY such that we can parallelize across parameter sets, for example, for parameter sweeps. We can parallelize across inputs for training on large-scale data sets. And we can parallelize across cells so as to simulate and train large-scale networks. Now there's one more thing, right? We can now actually perform gradient descent, but doing so can be much more challenging than training, let's say, standard deep neural networks. In particular, you can imagine there's lots of local minima. You can imagine parameters having vastly different scales. And thirdly, you can imagine that these kinds of biophysical simulations have computational graphs of many terabytes. We've developed a few utilities to overcome this. In particular, we're reparameterizing parameters, we're using particular optimizers, and we're using multi-level recursive checkpointing to overcome the memory consumption of backpropagation of error. Now, on the next couple of slides, I'm going to show you two examples that demonstrate that this then actually works and allows us to train biophysical network models. I'm going to show you two tasks. The first one is going to demonstrate that we can fit network models to data, in that particular case to calcium data. And in the second part, I'm going to demonstrate that we can use this kind of framework to train biophysical network models to solve computational tasks, obviously on MNIST. So let's get started with fitting neuron models to calcium recordings. We aimed to build a model of the mouse retina. The mouse retina processes light by first absorbing it in photoreceptors, which then pass on the signal to bipolar cells, which again pass it on to retinal ganglion cells, which are the neurons that relay the signals into the central brain of the mice. And our collaborators in Tübingen have recorded calcium signals or activity signals from these exact retinal ganglion cells. And they didn't do this just from the soma or from the cell body of these cells, but from every kind of dendrite on these cells. And they thereby found that these cells compartmentalize their response. We aim to build a computational model of this. We aim to build a computational model that is fitted to this data, so that is stimulated by random noise images and then predicts the calcium data of these dendrites. And to do so, we've built a hybrid model of this retina. We've simplified everything that's not completely essential to our model. In particular, for example, for the photoreceptors, we used a linear filter, and for the bipolar cells, we used single neurons with a nonlinearity. But we used full biophysical and morphological details for those cells which actually do matter to us, namely the retinal ganglion cells. The resulting network had about 600 parameters, and just to give you an impression, gradient-free methods usually struggle with about 10, 20, 30 parameters, right? So this is vastly more than what would be possible without gradients. And we're fitting these parameters based on 15,000 pairs of random noise stimuli and calcium recordings. And one beautiful thing about having a biophysical model here is that we actually don't have to worry too much about how we obtain a calcium signal from neural activity. A biophysical model just includes calcium channels, which inherently model calcium concentration and which thereby inherently predict how much calcium response there would be. And we just feed this into a mean absolute error function and then train on this. After training, the hybrid model or our computational model fitted data across multiple dendritic sites with a correlation of something between 0.1 and 0.5. And we then queried whether this trained model actually reproduces measurements that have been made based or in the actual mouse. For example, we've stimulated particular sites of this neuron, and we looked at how the signal propagates through the cell. And we found exactly what they found in experiments, namely that the signal compartmentalizes so that it doesn't propagate throughout the entire cell. We further quantified this by computing receptive fields. These sorts of fields tell us where exactly we have to stimulate or shine light in order to elicit responses in particular parts of the cell. And we found that these receptive cells on the left here roughly match those also made or also found in experiments. Now, I hope that this convinces you that differentiable simulation and gradient descent makes it possible to fit network models to data. In the second part, we're going to turn to tasks, to computational tasks. We've built a biophysical network model and trained it to classify MNIST. This network consists of about 800 neurons, and we're training single cell properties as well as synaptic properties for a total of about 100,000 parameters here. And we're fitting this to MNIST, which contains about 60,000 images. And I really want to highlight that I don't really believe that neural systems really classify MNIST, right? It's really just a benchmark that's showing what's possible, and that if one has a task that it might actually be realistic for the brain, then these kinds of scales are possible now. 100,000 parameters and large-scale data sets of about 60,000 images. Now, after training, upon being stimulated by a zero, the activity propagates through this network and then predicts a higher voltage in the output neuron corresponding to the zero, as compared to all the other digits, meaning that this was classified correctly. The network achieves an accuracy of about 95%, which is higher than an optimal linear classifier. And I want to highlight here that this network doesn't contain any kinds of artificial nonlinearities, right? There's no ReLUs or anything like that. So the reason this can even be better than a linear classifier is that the network just learned to use the existing nonlinearities, for example, of the sodium spike or of the synaptic dynamics, in order to improve performance. We can then also further study this network, and we find that, for example, the respective tuning of parameters is extremely important for task performance, and that just relying on the mean and standard deviation of parameters doesn't or isn't enough to perform this task. And we can do fun things such as adversarial attacks on these biophysical network models, again by the ability of Jaxley to compute the gradient. Beyond this task, we've also applied Jaxley to a bunch of other models, for example, to fit single neuron models to intracellular recordings, or to train biophysical recurrent neural networks to solve working memory tasks. You might now be wondering, how far can we take this? Very roughly, the largest system we can run on a single GPU contains about 2,000 neurons at full morphological and biophysical detail. At this scale, it takes about 2 minutes to compute the gradient, which is certainly not little, but which could actually train within a couple of weeks. And coming back to the original first slides that I've shown you, right? The fruit fly consists of about 100,000 neurons, meaning that it might be possible to simulate the entire brain of such an animal on about 50 GPUs and to train it, for example, to perform motor tasks, to fly, to walk, and to actually act in the world. In order to make all of these things possible, we've also ensured that Jaxley is an easy-to-use simulator that allows scientists to really customize their model, to insert ion channels where they want, to define new ion channels, to define new synapses, and really build models in a fully customized way. Now, to summarize this, I've shown you that biophysical simulations are a really powerful framework for building artificial neural networks, but that we have to infer their parameters from data. I've demonstrated two approaches for this. In the first part, I've talked about simulation-based Bayesian inference with neural networks, which are great because they are so generic, right? They can be applied to any kind of black box simulator for which we can easily run simulations. In the second part, I've talked about differentiable simulation, which I've shown you opens up new possibilities for scaling to more parameters, larger models, and larger data sets. Overall, I think it's a really exciting time for computational neuroscientists, because it's really a time where we have data that was just absolutely unavailable before, right? We have models of how exactly neurons connect, how exactly neurons look, and we have these models at a scale where they really allow us to simulate entire brains of animals. Biophysics allows us to incorporate exactly these things that are known about systems, such as connectivity, morphology, and ion channel dynamics into models. And inference allows us to then infer the free parameters from additional recordings of neural activity, or such that the system performs a computational task. And taken together, I hope that this will lead to a new generation of models in neuroscience that are actually plausible in a biological way, but also perform tasks as machine learning kind of models do now. There's a lot of people that were involved in this. First off, I want to thank Jan and Richard on the left here, who have really contributed a lot to the SBI toolbox, to methods on SBI, and to making these tools usable, flexible, and actually also useful to the community. There's also many people that have been involved in building, actually in training the kinds of models that I've shown today. And I want to thank Jakob and Pedro, who have been my supervisors throughout my PhD, as well as Axel Borst, who's now my postdoc supervisor. And finally, I want to thank the organizers of this conference for a great conference so far and for giving me the chance to talk here. Thank you all.
Thank you very much for a very fascinating talk. It's very interesting how you can approach this system. Do we have questions from the auditorium? Okay, please.
Thank you very much. So I'm mostly interested in how you kind of take a network which did not want to train and how you made it work. Is there something more general to be said about new types of networks? And like, did you learn anything that is slightly more universal than kind of your particular application? Thank you.
I think the ultimate thing that makes it really hard is unrolling in time, right? So that the more time steps there are, the more you will run into exploding and vanishing gradients. I think that's the ultimate thing for me. If you have a relatively short simulation, it's going to be relatively easy. If the simulation is super long, it's going to be relatively hard. What I often realize, or what I often saw, is that the norm of the gradient varies gigantically, especially in these long simulations, which makes sense, right? And so actually one of these optimizers that I'm using, they correct for this. They basically divide the gradient by its norm, thereby leading to gradients being just roughly standardized, and this helped a lot. Beyond that, I think the tools like parameter standardization and like reparameterizing, as well as multi-level checkpointing, all of these things are super important for training any kind of differentiable simulator. Outside of that, I wouldn't be sure if I want to recommend anything really. Yeah, sure.
Okay, thank you. Other people?
Yeah, thank you for the great talk. So I think during your talk, you mainly focused on simulating an artificial system that basically carries over all these properties of a biological system. But in your title of your talk, you refer to mechanistic models. And I'm wondering how you connect the dots there. So what you actually mean by mechanistic models in your title?
Yeah, so by mechanistic models, I mean models that really include the mechanisms we believe to be at play there, right? And that is biophysical models. We believe that there's a sodium channel that opens and inactivates, and that's models that include these exact mechanisms. So I basically use biophysical models and mechanistic models as synonyms, yeah.
Okay, thank you for your talk. So I was wondering how you did deal with a non-differentiable function like a sail or flow around. Did you use like state through estimation to estimate the gradient, or what exactly?
So by and large, we don't need it, because by and large, these simulations are just fully differentiable. They're fully continuous, and there's nothing to be done there. In the few cases where there are synapses that have some kind of properties, like for example, if you're into neuroscience, NMDA synapses have this magnesium block, which is basically discrete. In those cases, we use relaxation techniques or straight through estimation, but by and large, we don't need this.
How do you see the end goal of your research? I mean, is it to replace the current tensor-based computation by these biophysics-like simulations, or rather to model live systems using your approach?
I think both would be possible, right? I do think there is a chance that these kinds of things inspire, at least to some degree, how brains have achieved, for example, their energy efficiency, right? With knowing, for example, the exact connectivity matrix, one could imagine that they obtain some kind of visual processing with the minimum amount or with a minimum number of neurons. to me, to be honest, it's much more interesting to actually understand how brains do it. And it's interesting to me how exactly this detail or this hardware, as we heard in the beginning, of brains implements the computational functions of cells, and how we can use computational models to understand this better, to suggest hypotheses, to suggest experiments, and to understand this.
Okay. I think maybe my last question, basically, so you will, for example, now modulate brain of this fly. So do you see the consciousness? Because maybe, yes, this fly will be now reproducing somehow a real one. So what do you think about? Are we approaching to this ethic question? Now you will have a live fly, and this should be object which should be like, with consciousness, like, normal one. So do you think by this model you can go to the ethical questions that basically will reproduce so well living systems? It will be like taking this.
I hope so, right? And I mean, people are really working on all fronts of this. I am mostly working on just really building these tools, but people are working on like building models of the brain, building computations of the brain, and also like scaling this onto muscles, and then actually having like simulators that run around, like that make the fly run around in environments and make it fly around. So I think in a way, all the ingredients are to some degree there. The question will be how well it works to put these things together and how useful the resulting model will actually be. But I really hope that this is going to be possible soon. Yeah.
Okay, thank you very much for a fascinating talk.